So what is quorum sensing? So quorum sensing is a method of the communication. So signaling process is also one type of the communication method in a bacterial cell also via hormonal signal mechanism. But in case of the quorum sensing in a bacteria, so quorum sensing is a method of the communication between the bacteria and the another unable to the coordination of the group based behavior, or it also based on the population density. So basically quorum sensing is a communication method between the bacteria, how bacteria will be communicate with each other. So this is the method in which bacteria will be communicate with each other, and then it will be change in a behavior, or it also change in a popular density. So in this case, a quorum sensing method are communication between the bacteria, and then it will be coordinate, or it will be pass the message in between the bacterial behavior, or it also related to the population. So in this case of the condition of this bacteria, so in this bacteria they are the part of the communication mechanism. So the signal can change via gene expression. So this signal will be changed via gene expression, such as a switching between the flagella gene, or it also change into the gene like pili for the development of the biofilm. So in this condition, they are the condition in which a bacteria can be communicate, or it will be express the role of this express the role in a behavior like signaling molecule via gene expression, or activate the gene, or it also switching the condition of the particular flagellal condition. So this flagellal condition are also carried out via pili formation or the development of the biofilm mechanism. So these are the process of the quorum sensing mechanism. Quorum sensing mechanisms are carried out via signaling molecules. So what are the main signaling molecules are there? So signaling molecules are N-acetyl homoserine lactone. So these are the one of the signal molecules are secreted into the gram-negative bacteria N-acyl homoserine lactone. So this kind of the signal molecule are the class of the signaling molecule, or it will be involved into the bacterial quorum sensing mechanism. So this is the main molecule are involved into the signal from the bacteria to bacteria, or it also communicate with each other via secretion of the hormonal signal, or is like as a N-acetyl homoserine lactone. So in this condition, they are the class of the signaling molecule, and it also involved into the bacterial quorum sensing mechanism. So quorum sensing. So we are talking about the gram-negative bacteria or gram-positive bacteria. So the first identify the quorum sensing bacteria in the first system, it was be described in a vibrio fischeri. So this vibrio fischeri are the type of the bacteria in which they are the described by the Nelson and Hastings in 1979. This is the vibrio fischeri bacteria. These are the green fluorescent light will be emitted. So it's a symbiotic species, and that provides its marine eukaryotic host with a providing the light. So these are the fluorescent light producing bacteria, and this is also known as a one kind of the symbiotic mechanisms are carried out with the marine eukaryotes. So this kind of the bacteria will be involved into the quorum sensing mechanisms. His name is a vibrio fischeri. So quorum sensing are carried out in a gram negative bacteria, or in case of the gram positive bacteria. So these gram-negative bacteria are carried out via two gene expression. First is a LUX1, LUXL and LUXR gene. So these are the two genes are responsible for the generation of the quorum sensing mechanism or via gene expression, or in case of the positive gram positive bacteria, it will be involved into the peptide mediated quorum sensing mechanism. So these are the two types of the quorum sensing are carried out. First is a gram negative bacteria and second one is a gram positive bacteria. Gram negative bacteria are activated the gene expression via two gene LUXL and LUXR, or in case of the gram positive bacteria it will be involved through the peptide mediated quorum sensing mechanism. Quorum sensing mechanisms are also involved into the bacterial. It will be showing so it will be showing the bacteria can be showing the quorum sensing, or it will be required two things. First is a signaling molecules called autoinducers or the pheromones, or second one it is a receptor mediated and then it will be specially detected the signal molecule. So these are the bacteria can be showing a quorum sensing are required the two things. First things it will be a signaling molecule called autoinducer or pheromones will be induce the some kind of the quorum sensing activity, and the other one is a receptor can interact with the specific molecule or signal molecule and then it will be detect the signal and then it will be respond on the basis of this message can be received via receptor. So these are the autoinducer or pheromones mediated, or it will be a receptor mediated signaling can be received via bacteria and then it will be react on the basis of this signal molecule. So these are the example of the gram negative single species bacteria carried out in a quorum sensing mechanism. So first of all, quorum sensing mechanisms in case of the lower density. So this is the cell. This kind of the structure are known as a acyl homoserine lactose. So these are the autoinducer we have discussed. These are the signal molecule. This is the activator protein. These are the chromosome or acyl homoserine synthase. So these are the enzyme. So these are the 4 to 5 components are involved into the quorum sensing mechanism. So first of all, bacteria having the low density. It means 2 to 3 bacteria are available at the site of the reaction. When this bacteria can secrete the particular hormones or signal like a acyl homoserine lactose. So by secreting of this component, it will be populated at the particular site. So these are the population of this cell of the gram negative bacteria will be increased via releasing of this signaling molecule. So it will be converted from the low density to high density molecule. So this high density will be created when it will be sensitize the acyl homoserine lactone at the particular site, and then after it will be also secreted via further it will be secreted via acyl homoserine signaling molecule. So this is become a clump to each other and then it will be form the biofilm at the particular site. So first condition, bacteria having the low density via secreting of this signaling molecule, so the concentration of the bacteria will be converted into the high density molecule. Then after the further it will be secreted the hormonal signaling, then it will be clumped to each other and attract each other via this message. So it will be form the clumping to each other and then it will be form the biofilm. So these are the closer look inside the cell. So how the closer look will be generated? So first these are the homoserine signal molecule. So this signal molecule will be diffused through the cell membrane. So after all, a cell membrane will be diffuse this molecule, then it will be attached to then activate the regulatory protein. So this homoserine can be activate the regulatory protein, and after all, regulatory protein can bind with the DNA molecule, and then this DNA molecule can create the new acyl homoserine lactone. So in this condition, this enzyme are synthesize the acyl homoserine synthase. So this enzyme will be activated and it will be secreted the large amount of the acyl homoserine lactone as a inducer molecule, and the process will be repeated continuously. So number of acyl homoserine lactose will be increases in case of the cell to cell communication mechanism. So these are the reason behind this, the all the bacteria how it will be closed loop inside the cell. First, acyl homoserine lactose will be induced via particular bacterial cell, then it will be interact with the other bacteria can activate the regulatory protein interact with the DNA can induce the acyl homoserine synthase enzyme. So this enzyme will be activated and produce the another kind of the or other acyl homoserine lactose in a large number. So this process will be continuously repeated. So this the amount of this acyl homoserine lactose will be increases, and the it will be interact to the other bacteria, and then it will be form the biofilm. Okay, so this is the main process of the acyl homoserine lactone. All of you clear? All of you clear this mechanism? Low density to high density, and high density are activate this process, and then it will be converted into the biofilm. Okay, so this is the mechanism behind the gram negative bacteria of the biofilm formation or induce the bacterial communication. So these are the two genes we are all discussing. Two genes are responsible for the converting this quorum sensing mechanism, LUXL or LUXR. So this autoinducer of the acyl homoserine lactone or N-acetyl homoserine lactones. So these are the two autoinducers are play important role in a quorum sensing mechanism. So it is a synthesized by the LUXL homologous gene or LUXR homologous gene. So these are the two genes are responsible for the generation of the autoinducer, and then it will be produce the communication mechanism between the two cell. So first LUXL. So these are the LUXL can produce the acyl homoserine lactone. So these are the transcription is not activated due to the low cell density, because these are the one gene can activated, and then it will be only one molecule will be synthesized, or this molecule cannot be bind with the particular receptor or particular targeted gene. So these are the gene cannot bind with the acyl homoserine lactone, and the transcription are not activated. So these are the transcription is not activated due to the low cell density molecule. After all, a transcription is activated and it will be converted into the high cell density. In case of the LUXL will be activated acyl homoserine lactone, and this acyl homoserine lactone are increase in amount, and then it will be bind to the targeted gene. So this targeted gene are activated the other gene like LUXR, this one, and this LUXR can bind with the acyl homoserine lactone, and then transcription will be activated and the gene will be expressed, and then it will be synthesized the proteinous part. So these are the gene mechanism when the low density acyl homoserine lactone cannot be interact with the particular acyl homoserine synthase enzyme, and these enzymes are not capable to transcribe the particular targeted gene. But in case of the interaction with the particular acyl homoserine lactone, so this condition will be activate the particular targeted gene, and this gene will be activated, and then it will be synthesize the protein via transcription mechanism. Okay, so all of you clear? LUXL and LUXR, these are the main gene are responsible for the formation of acyl homoserine lactones as a inducer, and then after it will be activate the other bacterial cell, or it will be induce the cell population or cell density at the particular site. Okay, so can we move further? Bioluminescence. So this is the condition in which a vibrio fischeri can produce the light via activation of the these two gene we are discussing in our next slide, LUXL or LUXR. So these are the bioluminescence. Via activation of this gene, it will be synthesize the protein, and this protein are responsible for the generation of the light. So this bioluminescence is defined as the emission of the visible light from the living organism. So these are the living organisms. Vibrio fischeri can activated via these two gene application, or via application of these two gene, it will be produce the bioluminescence, or it will be known as a visible light via living organism. So these bacteria are the most numerous of the light emitting form, and this light emitting via carried out is one of the most widely studied in bacteria, vibrio fischeri. So we are discussing in first slide. So these are the product of this gene, and these are the vibrio fischeri. So this vibrio fischeri can activate it and produce the light, visible light. So these are the green color is a fluorescent light. It will be activated the particular bacteria, and then it will be produce the light. So these are the fluorescent light is a product of the particular gene activation of LUXL or LUXR. Okay, so what is the importance of this quorum sensing mechanism? So importance is a one type of the bioluminescence is a production of the light in case of the bacteria. Exopolysaccharide productions are also a part of the quorum sensing. Virulence part is also a part of the quorum sensing. Virulence, it means is a poisonous condition to the other animal. Then conjugate or plasmid transfer mechanism, antibiotic production, exoenzyme production, biofilm production. We also discuss in biofilm production via aggregate the particular bacteria at the particular site. So these are the concentrate at the site of the one site of the bacteria, and then it will be formed the biofilm in the particular area. So these are also a application of the quorum sensing mechanism. And the last one is the growth inhibitor, also important characters of the quorum sensing. So these are the 7 to 8 characters are involved into the quorum sensing mechanism: bioluminescence, exopolysaccharide production, virulence, conjugate plasmid transfer, antibiotic production, exoenzyme production, biofilm formation, or growth inhibitor. So these are the application of the quorum sensing mechanism.